So we're starting a new topic now, which is the amino acids. Amino acids are, in a very literal sense, the building blocks of life. You may have earlier, when you were younger, studied of a group of compounds known as proteins. Proteins, which took that same accolade. In fact, the word protein comes from the Greek word proteus, protes, which means foremost or of most importance. So proteus means of most importance, and a protein is basically those molecules, those biomolecules which were deemed to be the most important. They are the scaffolding of everything. Nucleic acids fold in specific conformations, entwined, enmeshed in the most compact way possible, the most compact form of data storage known to mankind, because they are assisted by proteins. Nutrients are absorbed into cells and waste released from them because of channel gate proteins. The delicate balance, the delicate ionic balance between sodium and potassium in a neural channel and in the plasm surrounding a neural channel is also maintained by proteins. The very way we think is because of a flow of ions in electric currents, which are regulated by proteins. Not to mention that proteins are also the major structural motif of any being, from plants, animals, bacteria, anything. All of us have this much in common, that we are based off of proteins. Because proteins are some of the most complex heteropolymers there are. We've studied some simple homopolymers so far. Cellulose and amylose, all of which are glucans, consisting of just one monomer repeating ad infinitum. These guys have varied. Proteins are made out of 20 to 22 amino acids, depending upon which species you are. Almost all species, I think all species that I'm familiar with, have the basic 20 amino acids, and there are an additional 2 that can come and go. And these are what we call the proteinogenic. Proteinogenic amino acids. Amino acids from which proteins are built. But there are so many more. There is GABA, for instance. GABA, or gamma aminobutyric acid, is a neurotransmitter which is essential to the way we think and feel. It's part of the way our brains work, and without it we would be very different beings indeed. It is also an amino acid. There are amino acids which are part of this 20 to 22 proteinogenic amino acids. They're not amongst the protein building amino acids, and yet they're still part of proteins. Because after you assemble the protein out of these 20 to 22 proteinogenic amino acids, sometimes there are biological processes that modify the constituents, that modify the monomers after polymerization. So you can see that amino acids have a very rich wealth of importance in biological systems. They make up the structural motif through ordinary structural proteins. They make up the active catalytic motifs through enzymes, because they are the major catalysts of everything going on in the cell. They make up control and recognition, because they are the parts of the receptors that engage with any other molecule. They're what allow cells to acknowledge the arrival of hormones. Okay, they're what allow cells to switch on and off tiny currents. These are what allow the plants to use the proton current that they derive from the photolysis of water and convert that into ATP, because they have tiny motor proteins that work exactly like motors that run off a proton current instead of an electron one. That is the wealth of power that proteins have. They are more fundamental to life than anything else we know, and therefore their constituents, the amino acids, are one of the most important topics we're going to study till date. Not only are they important to biology, but they're becoming increasingly relevant in a field that is known as supramolecular. Where do I write this? Supramolecular chemistry. Supramolecular chemistry is the chemistry at the level above the molecule. Quite some time back, I think in the 70s or maybe even earlier, there were various scientists like Jean-Marie Lin who were studying how Proteins can tear sodium and potassium ions apart. I said that that's important because the exchange of these sodium and potassium ions is what keeps the current in our neurons flowing. And he realized that what happens is that proteins actually, along with other various biomolecules, get together at a scale greater than the molecule. Molecules fit in with each other, they lock up together, they form temporary associations via hydrogen bonding and van der Waals forces and coordinate bonding. And these are very large, huge associations of many, many molecules. And proteins are the major structural motif of these supramolecular assemblages. This allows them to carry out a remarkable number of functions. Again, enzymatic catalysis is one of them, but also a variety of transport. For instance, hemoglobin is a very important protein, and it binds reversibly to oxygen and transports it all around the body. Last year's Nobel Prize in Chemistry was given to scientists who were using these concepts of biochemistry. For instance, directed evolution, where they were basically, how do I put this? They had a group of bacteria that were evolving, and these bacteria, like all organisms evolve, as you know. And what they would do is they would kill off all the bacteria that did not make an enzyme useful to humans. So say they wanted an enzyme to oxidize something, and then the bacteria developed and some of them mutated. Some of them had an enzyme that was better at oxidizing, some of them had one that was worse at oxidizing. So they killed off the ones that were bad at oxidizing, and then what ended up happening is that you had these bacteria producing this really good oxidative enzyme. In a similar manner, you can get more and more specific enzymes, and you can get the best catalysts in the world out of using them. And those catalysts, those enzymes, are built, as I've said before, out of amino acids. So coming, as I said, to some specifics about amino acids, we have to see what they really are chemically, what their fundamental constitution is, since I've waxed eloquent about their properties and their uses. So amino acids is actually a shorthand term. It stands for amino carboxylic acid. Okay, so that means it bears an amino group, that is an NH2 somewhere in its structure, and bears a carboxylic acid, a COOH, somewhere in its structure. Okay, so that's all that this is telling you. It's got an amino group and a carboxylic acid group, right? So that could be any structure, any arbitrary structure like this, which has a carboxylic acid group and an amino group, right? This is a possibility, right? Also, so is this a possibility? Or even one where the amino group is here. This is also a possibility. So the amino group and the carboxylic acid group can be any length apart. However, when we come to what they really are, natural amino acids are all L-alpha amino acids. That's what this point over here says. So the alpha here is what I want to draw your attention to. We'll come back to the L over here later. Don't worry, it's not as bad as carbohydrates could sometimes get. But when we say alpha amino acid, we simply mean that the carbon attached to the COOH is also the carbon attached to the NH2. This would be an alpha amino acid. If it was here, it would be a beta amino acid. Here we'll make it a gamma amino acid. Here we'll make it a delta amino acid. So you have alpha, beta, gamma, delta, the first four letters of the Greek alphabet. And natural amino acids contain the NH2 at the alpha position. So it's an alpha amino acid or an alpha amino carboxylic acid. Let's come to the next bit. It says natural amino acids are all L-alpha amino acids. So what do we mean by L over here? So we know that they're alpha, so let's look at the structure over here. There will be an NH2, a C, a COOH, and then two other arbitrary groups. In proteins, the amino acids are made up proteins, the proteinogenic amino acids we spoke about earlier. You're going to have one of these groups being a hydrogen, and the other one can vary. And it's this other one that we're most interested in. This other one is what helps us tell, is the only thing that tells actually the different amino acids apart from each other. So it's the identity of this other group over here. There is one particular amino acid called serine. Okay, now I should note straight up here that most of the amino acids end in the letters I and E, and I pronounce them amino acid and I call them serine, valine, threonine, and so forth. But there are also people who pronounce it as valine, serine, amino acid, and so forth. The choice is yours, but it's pronounced either way. So if you look at the structure of serine, you can see that it very closely resembles a carbohydrate. This is essentially a carbohydrate with the aldehyde oxidized, the OH here replaced with an NH2. That's all that's happened. That's the only way it differs from a carbohydrate. So we can apply our D and L nomenclature. Basically, we can say, look at serine and then tell me, is the NH2 on the left, then it's L. Is the NH2 on the right, then it's D. And we can transfer this logic back to any amino acid, regardless of whether its R group makes it resemble a carbohydrate or not. So this D and L logic that we define for carbohydrates in terms of glyceraldehyde is defined for amino acids in terms of serine. And the logic is very simple. Is the NH2 on the left or on the right, provided the Fisher projection is arranged in this manner. COH on top, then C, and then the R group with the H on this side. Okay, now the next thing we ought to know is what the variability of this R group is like. We'll cover this in a lot of detail right now, but what I want you to understand is that the R group is not limited to the typical organic R group. In organic chemistry, we generally use R to mean an alkyl group, or at most an alkyl/aryl group, one of the two. But in amino acid and protein structures, R is used to refer to literally anything you could think of. So there could be, there'll always be at least one carbon, there'll be some carbon chain. But on that carbon chain, there can be all kinds of functional groups attached, including just aryl groups or OHs, other acidic and other amino groups. There are all kinds of groups that can feature on this R. So I want you to remember that it's not just an alkyl chain we're talking about. This will have some functional groups on it, and it will have some reactivity. Finally, I want to clarify what I mean here by natural amino acids before we move on to the next bit. What is a natural amino acid here? So when I say natural here, I am referring to the amino acids that are found in living organisms, but I'm also making a secondary assumption. I'm referring to the ones that are actually used to make proteins, i.e. the proteinogenic amino acids. There are, for instance, some D amino acids that are used by bacteria in a defensive coat around themselves. I'd encourage you to pause the video and try and think of any reason why bacteria would try and use D amino acids to protect themselves. Okay, I hope you've given it a good go. Essentially, a D amino acid, being something that's unnatural or something that's not found in the proteins that organisms typically digest, means that this D amino acid is going to be impossible for any predatory organism to digest. So the bacterium is quite safe. There's no enzyme that any living system has evolved to degrade D amino acids so that it can go and kill these particular bacteria say, because D amino acids aren't part of the food they eat, so they never had to need to degrade them. They never had to need to work with them. Their enzymes are not enzymes that fit with D amino acids. We'll see that when we talk about enzyme fit in a later part of this video. So we've covered what these amino acids chemically are, that they're amino carboxylic acids. We've covered that they're typically L-alpha. We've said what the L means by comparing it to serine, which in turn we compared to glyceraldehyde and other carbohydrates. And we've also said what the alpha means by talking about the position of the NH2 on the structure. The last thing I want you to realize is that there is a bond known as a peptide bond that links these amino acids together and forms a protein out of them. Essentially what happens in this particular case is very simple. It's you have this R, COOH, right? Here the R isn't the R, okay, let's just call this A for amino acids. So A, COOH and NH2, right? So the amino acid has got NH2 and COOH. And then there's another, let's call this A dash, another amino acid with its own NH2 and COOH. So basically all that happens here is that this COOH and let me write the H on this side. One of these H's, they go out as water. This goes out as water, and you're going to be left with an amide group like this. C double bond O NH. We're going to study the nature of the peptide bond in a great deal more detail. I just wanted to cover this link between amino acids and proteins in the briefest of things so that you don't lose track of what we're studying amino acids for, to understand the polymers, which are proteins. Right, so in the previous video we've covered different characteristics of amino acids, the simplest of course. We've covered what their basic structure is like. We know that they are all L-alpha amino acids, at least the proteinogenic ones. We've covered also some of the amazing things that their polymers, namely proteins, are capable of. How they learn structure, how they catalyze reactions, all sorts of things. But now it's time for us to come down to a very specific chemical property that they have. That is, they exhibit dipolar forms. So we're going to understand what those forms are. And then we're going to understand a technique known as electrophoresis that makes use of this chemical property to segregate amino acids, to separate them so that we can study them individually and not just as a category of compounds. So what is a dipolar form? Now a dipolar form is exactly what it sounds like. It's something that has two different poles or two different polarities on it. There is another word that is more common for dipolar form that you may well have found. Dipolar form or dipolar ion. This other word is a Zwitter ion. Zwitter ion. Zwitter is the German word for hermaphrodite or transgendered person, someone between the sexes. And the Zwitter ion is something that is between a cation and an anion. Namely, it is a single covalently linked compound or molecule which bears a positive charge in one part of the molecule and a negative charge in another part of the molecule, and so it is overall neutral. Let's see how amino acids achieve this. So this is the typical structure of an amino acid that we have already studied, right? Let's check everything we know. First, it should be an alpha amino acid. Recall that alpha in amino acid has nothing to do with alpha and beta in terms of carbohydrates. These are not terms used to nomenclate anomers. Here alpha, beta, gamma, etc. simply tell us how far apart the NH2 and the COOH are. Here is my COOH group. Here is my NH2 group, and they are one carbon apart. So this is an alpha amino acid. This is an alpha amino acid that we've checked. Next, it has to be an L alpha amino acid. L means that in this particular case, the NH2 has to be on the left hand side. Recall that we derived this logic for L and D by considering a specific amino acid called serine, in which case this R group made the whole thing appear rather a lot like a carbohydrate. So we were able to just transfer our DNN knowledge. So even when we use an arbitrary R group that doesn't make this overall chain resemble a carbohydrate at all, the logic still follows. If COOH is at the top and R at the bottom in a Fisher projection of an alpha amino acid, then that alpha amino acid is termed L if in this frame NH2 is on the left, and it is termed D if in the same frame NH2 is on the right. So yes L, yes alpha, and it's an L alpha amino acid. Now let's study its different forms. Down here, this arrow indicates the variation in pH. Here we have what form it will assume at pH equal to 0. Here we have what form it will assume at pH equal to 14. Now let's see this. This is a COOH group, right? What happens when you put it in water? It dissociates and it becomes negatively charged. It releases H+. It becomes COO minus and the H+ is released. But and therefore the COO minus is what we call a conjugate base, right? It's a conjugate base. If the acidity around it is sufficiently high, it will reassociate back together. So let's say this H broke away when we dissolved it in water. Then we reduced the pH towards 0. Lots and lots of H pluses were nearby. This H plus came and joined back on. So in this particular form, this is going to remain. This is going to remain as is. It's going to remain COOH, because even if we removed a hydrogen, when we come back towards low pH, the hydrogen will come back and join on. On the other hand, this NH2 had a lone pair of electrons here. As the basicity decreases, or as the acidity increases towards 0, what's going to happen is that H plus ions from the solution will go and attack that over there. So if you have an H plus ion like this, let me draw it over here maybe. This is an H plus ion. It will come like this and attack the lone pair. And that's exactly what's happened here. An H plus ion has come, brought with it a total of a positive charge, and has converted this whole thing into a cation. It's a cation because this has been, the amino group has been protonated, but the COOH has not been deprotonated. Now let's bring it closer to pH 7, to the middle of the scale. Okay, in the middle of the scale, what's going to happen is that this NH3 plus will remain an NH3 plus, because if you put ammonia or an amine into ordinary water, it automatically acquires a proton. The Kb value is high enough that it automatically acquires a proton from just neutral water. It doesn't need to be highly acidic. So this becomes NH3 plus, but this COOH will lose that H plus and it will not come back and join back on, but will remain in solution. And so therefore this will become COO minus. And you can see there will be a resonant characteristic. You can't tell which oxygen is doubly bonded, which is singly bonded. This is just basic resonance. So this is very stable. Now here you can see that this overall complex is neutral. This is neutral. This is cationic. And if we were to go all the way to pH 14, the opposite would happen, right? There'd be so many OHs involved that even if this NH2 had become an NH3 plus, the OH minus would come and rob the NH3 plus of its H, right? If they have lots and lots of OH minus ions, they would come and they would remove any H plus before it can come and stick onto this NH2, and they would also remove any H's from here. So this would be neutral, this would be negative, so overall it would be anionic. The cool property of amino acids is that in this neutral zone, when it is not too acidic nor too basic, at physiological pH, in solution, the amino acids are zwitterionic. Zwitterionic. And this zwitterionic property is extremely important. One very common question that you were asked actually is that if you separate amino acids from water, what form do they take? Will the amino acids form a liquid, or will they be liberated as a gas, or what? And the answer is, at physiological pH, they will be solid. They will have these strong ionic forces. So if you try and somehow condense them, say you cool it down or something so it precipitates out, then these will act as ions. They will attract each other. Now you might say, but each of these amino acids is not positively charged or negatively charged, right? So what polarity will it attract? And the answer is it doesn't matter. It's a bit similar to how when water crystallizes, all that matters is that there is some dipole moment. Now here the dipole moment is much higher because there are actual ions in the amino acid. There are actually charged groups, not just polar covalent bonds like in water. So there will be a strong dipole attraction, and these dipolar ions or these Zwitter ions will come together and form an ionic crystal. So one very common question, what is the state of amino acid? Solid, liquid, gas? Mark solid always. Except when they dissolve. In solution phase is different. But when you try to remove them from the solution phase and study them independently, if you crystallize them from water at neutral pH, then the crystals will always, you'll have strong solid crystals because of the dipolar nature of these ions. Okay, now let's come to electrophoresis. Right, so we come now to the topic of electrophoresis. Electrophoresis is a phenomenon in which different molecules or different chemical species are separated on the basis of their charge. Okay, so in this particular case, what we're doing is something that is known as a gel phoresis. So the medium or the matrix through which the different chemical species are progressing when they are being separated is in this case a gel. So what gel? Generally when we are separating proteins like this, we use polyacrylamide. So polyacrylamide gel electrophoresis is often known as PAGE. So you will hear scientists, you will hear people working in biotech talking about doing a PAGE. This is basically what they mean. So in this case, we've taken all these different amino acids. Then there are many other gels that can be used. Please don't go by this. This is just a gel I thought I'd mention. Right, so electrophoresis can occur in any matrix, not just the gel that we spoke about, even in other forms. But what essentially electrophoresis involves is this. You take a comb-like structure and you prick it in the gel. So you prick the gel. Once you prick the gel, you get many tiny holes. These are called wells, like this, depressions in your gel. And what you do is, you take a micropipette and you put a little bit of your sample, whatever your sample may be, in each of these wells. Then you apply a positive charge from this side and a negative charge from this side. What will happen? They'll start to move. The amino acids will start to move in either direction, depending upon at the pH that you have set in the gel, are they positively charged or are they negatively charged? And you can see that they've moved in all directions. Now let's say I do this again. but I adjust the pH slightly. Now the pH is such a value that the last sample, okay, the amino acids of the last sample do not get charged at all, right? So then what will happen is that the amino acids of this sample will not move in either direction. They'll stay right over there. So you will know that this is the amino acid you're after, the one that has this particular isoelectronic point. Sorry, isoelectric point. Isoelectric point, which is symbolized by Pi. It is the pH at which the sample is overall neutral. That actually means that the majority of the sample is in a dipolar form. So like if you were to have a graph of cut that bit out. Majority of the sample is in the dipolar form. Now one way to use this to just to check if you already separated your amino acids based on some other method, you can check it is the right amino, that the separation has worked correctly by checking that whichever one you expect to be non-motile, whichever one you expect to be stationary at this particular pH is actually stationary. But more often what will happen is in the same well you will have multiple different amino acids, right? And so you will have some will have come out here, some may have come all the way here, some may have be here, but a little bit is stationary. And then this little bit which is stationary is the amino acid that you are after. It is the one that is neutral at that point. So this isoelectric point can be defined in two simple ways. One is net neutrality. So the sample is net neutral. Overall, no charge. Second way to define this is no movement. No movement during electrophoresis. Now we will move on to learning how to calculate the Pi, or how to calculate the isoelectric point, of any given amino acid. Right. So now we're going to try and calculate the isoelectronic point, or the Pi value, for a particular class of amino acids, and we'll come back to what I mean by that later. Okay. Just remember that the formula we're going to derive is not completely generic. This is one case of the formula, and I'll show you what the other cases might look like. Okay, so let me introduce the following notation. A minus is a completely deprotonated amino acid. Okay, so when I write A minus, I mean that it's NH2, not NH3 plus, and the COOH has become COO minus. This is completely deprotonated. This is what happens at a very basic, very alkaline, in a very alkaline medium. AH is neutral. If the sample is overall neutral, then a large proportion will be in the dipolar form. Some other smaller proportion might be in an uncharged form. But there will also be some tiny amount which is cationic and a tiny amount which is anionic. All we can say is that the two amounts must be equal. And it is on this equality that it is on the proposition of this equality that our derivation is going to be based. So let's start. AH is neutral, AH2 plus is protonated, A minus completely deprotonated. Now, this AH2 plus will exist in an equilibrium, right? It's going to tend, this is at the most acidic level, it's going to tend to deprotonate in some amount, and that is taken care of by this Ka1 value. Ka1 is the first acid dissociation constant. So this will dissociate and it will become H plus with AH. AH being completely neutral. H plus with AH being completely neutral. Right? Similarly, this AH will further be in equilibrium with the A minus, the completely deprotonated form. And here it will be losing another H plus. This is mediated by a different constant, or this is described by a different constant, Ka2. Okay. Now, what are we saying? We said that while the majority will be dipolar, and some will be uncharged, there will also be a tiny amount of cationic and a tiny amount of anionic form. And these two forms must be equal at isoelectronic point, because the overall sample is completely neutral, right? So, what we're going to do is we're going to calculate this cationic form. What is its concentration? In terms of Ka1 and H plus and AH. Then we're going to calculate the concentration of A minus in terms of Ka2, AH and H plus. And we're going to equate these two concentrations. And that way we'll be able to relate the concentration of H plus, which is basically the acidity, the pH, to the Ka1 and Ka2 values. So let's do that. AH2 plus. Now let's see how we can get this. So we know that Ka1 will be equal to the concentration of H plus into the concentration of AH divided by concentration of AH2 plus. If we transpose AH2 plus and Ka1, we get the following relation. We get AH2 plus is equal to AH plus AH all over Ka1, where all of these chemical species are not numbers, they are the concentrations of the respective species. Then when we come to A minus, if we do a similar thing over here, Ka2 is equal to this into this upon this concentrations, obviously. So again, if we do a transposition, we are going to get A minus, the concentration of this, is equal to Ka2 into AH by H plus. You can pause the video, get these values yourself and try and derive what the value of H plus should be. I encourage you to do this. Okay, I'm assuming you've tried. Hopefully what you've seen is, if we equate these two values, then this AH is going to get cancelled. So what you'll have is H plus by Ka1 is equal to Ka2 by H plus. Transposing, we get H plus whole square is equal to Ka1 times Ka2. Now, remember what we need is the pH, right? So, if I take a pH is basically taking a negative log, right? pH is taking a negative log. So, if I take P of this, then what I'll end up getting is 2 into pH is equal to pKa1 plus pKa2. This RHS will also be written as minus log of Ka1 minus log of Ka2, but minus log is P. So write it like this. This is 2 into pH, and this 2 can be transposed over here. So if we know in any given amino acid like this, in an acidic medium, so that both of these can be deprotonated. In a neutral medium, only one of them can be deprotonated. Then we'll have to write this as Ka2 divided by Kb1 and all kinds of things, just to keep the notation same by using Ka's everywhere. So what we're doing is we're going from 0 pH towards 14 pH, right? We're not trying to do it in the intermediate value. We're actually starting at near 0 pH and seeing what happens as you decrease the pH. Okay, so both of these can be deprotonated. This is NH3 plus, so this is tetravalent nitrogen. Obviously, it is going to be deprotonated. This is COOH. So these will both be deprotonated, H and R. If I am given this value describing this guy and this value describing this guy, then the arithmetic mean of these two is the PI value. This can be written as PI for isoelectronic point, because it is that specific pH where they are equal in concentration. Right, so here's a sample question for you guys. The sample question is as follows. This is a different kind of amino acid. It's different from the first kind because in the R group, I have another COOH. It has a pKa value, pKa3, just like this is pKa1 and this is pKa2, like we covered before. This is called aspartic acid. It's an acidic amino acid. By acidic amino acid, I mean that it actually has one acidic group and then it is two acidic groups and one amino group. So the number of acidic groups outnumber the number of alkaline groups. So in the case of this acidic amino acid, which is aspartic acid, you are asked to try and formulate the PI value in terms of these pK values. Okay, so please pause the video, give it an attempt, and then we'll start, and then I'll show you how to solve it. Please pause it now. So I assume you've paused the video and you've tried. Now let's see how we solve it. But first let's clarify, this is one very common misconception that people might have. People often think that because in the previous case, the PI value was the arithmetic mean of the two pK values. Here I'm dropping the A because we're only considering Ka, we're only considering acidic. I'm writing it as K1, K2 and K3, okay. So, in since Pi value was the arithmetic mean of pK1 and pK2, then when we have 3 pKs, it must be the arithmetic mean of these also. But that would be wrong. Why was this true? This was true because the concentration of H plus was the geometric mean of K1 and of K1 times K2. The geometric mean of K1 K2 of K1 and K2. Sorry. And when we take logarithms, or by exception you take logarithms and you multiply by -1, doesn't make a difference. When you take logarithms of geometric means, you might end up getting an arithmetic mean, right? So, if this is to be the case, then we must have the relationship of this being equal to the cube root of K1, K2, K3. K1, K2, K3. But there's a problem with this. If this has to be the cube root of this term, then H plus must exist as a cube. In some, you must be able to get the concentration of H plus cubed in one of your equilibrium equations. That is not going to be possible here. Because how did we get this H plus squared? We got it by equating two different concentrations. We equated the concentration in the cationic form to the concentration of the anionic form, right? And by making that equation, we were able to get somewhere. Here, if you won't be able to get all three H pluses on one side and the K1, K2, K3 on the other side, because we are only equating two concentrations. That's the definition of Zwitter ion, right? So again, remember this very clearly, when the Zwitter ion is maximum in concentration, then the cationic and anionic forms are equal in concentration. It's very hard to get an expression for the concentration of the Zwitter ion and then try and differentiate that and find what its maximum value will be. Much easier to remember when the Zwitter ion is at its maximum, cation and anion are at equal values, equate those and solve. So if you made this mistake, you can pause the video now and try again if you like. So having rubbed off that misconception that some of you might have had, it's now time for us to come to the actual formula for this. And remember, all we need to see is when the cationic and the anionic forms are equal in concentration. So let's begin by seeing something's wrong in this figure given to us, right? What's wrong? What's wrong is that a pKa1 is actually not defined for the amino group. I've given it to you like this because that's the way you'll find it in an exam. But that's actually incorrect, as you know. Amino groups are basic. So this is actually the pKa value of this whole thing when it is in the cationic state. Okay, that seems simple enough. Now, what are the four states that this is going to exist in? Okay, there are four states. There actually are four of them. So let's go over what the four states are. The first state that we're going to be concerned with is the cation. Okay, this is what we have over here, which is the entire H2N group like this. Okay, that seems simple enough. Then it will go to the Zwitter ion. Okay, as this gets released, then it will go to the anion once this is released and this is as it is. Okay, and then finally to the double anion. The double anion with a 2- charge. So if you're not quite able to see how it will achieve these four forms, don't worry, it's not that hard to do. Let's just take it step by step and see what happens as we go through each and every pKa value. Okay, so the first thing that's going to happen is that this entire H2N over here, let's look at the three different groups here and decide when they'll start losing these hydrogens that they have. Okay, so COOH, both of these COOHs, okay, they lose a hydrogen really easily. They lose a hydrogen really easily. Okay, so what's going to happen is they're going to lose the hydrogen first. Okay, but because NH2 is a strong base, we know that it tends to get an H plus quickly. It's not going to lose an H plus quite so quickly. When you start reverting back into a basic mode, okay, when we start reverting back towards basic pH, what's going to happen first? These are stronger acids that are going to react faster. They're going to react faster and they're going to lose their H pluses. By basic, I don't necessarily mean on that side of pH 7. Even as we approach pH 7, it gets more alkaline. On an absolute scale, it's acidic, but it's relatively alkaline compared to the most alkaline scale, right? So as we move upwards on the pH scale towards 7 and then beyond 7, what's going to happen is that the strongest acids are going to lose their hydrogens as fast as possible. But then to remove this guy, we're going to need a fairly strongish base, because NH2 itself is a strong base. That's why it's grabbed hold of the H+. It's going to need an even stronger base to remove the H+ from it. So it's going to have to go back into the other side. It's going to have to cross pH 7, because we know that pH 7, when we mix ammonia with water, it does hang on to the H+. So the order in which these are going to get released is first the 2 or the 3, either this COH or this COH is going to release the first proton. The other COH is going to release the second proton. Okay, so let's see what's happening. Here it's a cation. Okay, then one of the COHs releases a proton. Immediately it becomes a zwitterion, as we're familiar with. Then this CH3COH will release a proton. Okay, and now it's become an anion because it's got 2 minus charges and 1 positive charge, so it's an anion. Then finally, when you come to a double anion, what's going to happen is that this NH3 whole plus will release this H plus, and the H plus will go out and it will be left as plain old simple NH2. So what we have effectively is this order in which you release these H pluses from here and here first, and then this guy is at the very, very last. Okay. Now you can do out the equilibrium equations, and I encourage you to do it for yourself. I'm not going to do them out here, but you can do them out and it will help you. And you will see that what we basically need is the point at which the cation equals the anion. Now the cation to zwitterion is an equilibrium, and the anion to zwitterion is also, the zwitterion to anion is also an equilibrium, just as we saw before. We don't need to consider a double anion at all, because we need the point where these two concentrations are equal. When we take these two concentrations, we see that the cation to zwitterion was the loss of one of these COHs. Zwitterion to anion was the loss of the second COH. Only when it is a double anion do we have to worry about pKa1 at all. So we can do exactly the same logic. We get what I've done here, I've just written K2 and K3 instead of Ka2 and Ka3, because as I've already said, we always use Ka values just for ease of convenience. We actually do look at it from the most acidic point of view with an NH3 plus, just so that we can use only Ka values and we don't have to confuse ourselves by using Ka and Kb values. So I'm just using K1 and K2. We have this, and that implies, as we saw before, that pH is equal to pK2 plus pK3 by 2. And hopefully you can see how this is going to come, because we just have to write the equilibrium, this equilibrium equation, which is the K2 equation, and this, which is the K3 equation. Actually, these two could be flipped. It depends which of the two COHs will lose the hydrogen first, but both of them will definitely lose it before this guy. So we just need to take the average of these two pK values. By the way, if you're given a similar amino acid where the R group contains another amino group, okay, like say arginine, arginine, or lysine. Lysine is actually the best example. Arginine isn't an amine group, it's another basic group. So let's go with lysine. And I'll give you the structure a little bit later in the same lecture. So when we look at lysine, there's going to be an NH2 group over here. And then we just need to take the average of these two NH2 groups, the two pKa values, to get the Pi. We don't need to worry about the pK value of the carboxylic acid at that point in time, because again you will get exactly the same thing. You will have double cation to zwitterion to anion. So again we'll want the thing between cation and anion. So we'll ignore the double cation side, and the double cation side always the first. Let me write this properly. So I'll have 1 second. Let me switch color. So switching color of the chalk, the first thing I'll have is a double cation. So this double cation will again be in equilibrium with this cation, and the double anion will not exist, because we're talking about something with an NH2 group here and not a CH3COO minus. So again, the first guy The first guy to lose a proton will always be a carboxylic acid. Okay, and we will always need the equilibrium between the cation and the anion. So we'll ignore the carboxylic acid and we'll average the two pKa values of the two amino groups. So whenever you have two groups of the same kind of an amino acid, average their pKa values and you will get the pi value, and you can neglect the other one comfortably. Only in the case, which actually the majority of cases, but only in those cases where you have pK values which are equal, when you have equal numbers of the amino groups and the carboxylic group, basically 1 and 1, do you need to consider both the amino and the carboxylic. Otherwise, whichever one occurs twice, you take the average of those two pK values and you will have the pi value. And now we can move on. Right, so now let's look at the different types of amino acids there are. Okay, there are 20 basic amino acids. By basic, I don't mean alkaline. There are 20 fundamental or 20 elementary proteinogenic amino acids that are found across all living organisms. There are 22 total proteinogenic amino acids. By this I mean 20 of these amino acids are common to all living organisms, and depending upon the species, they may have up to 2 extra. For example, human beings, we have 21 amino acids. We also have an amino acid called selenocysteine, which is not in this list. But these 20 are common to all living organisms. So we're going to be talking about and studying these 20 for now. Now, these 20 can be divided further into 4 major types. The non-polar amino acids, the polar amino acids, the acidic amino acids, and the basic amino acids. And this differentiation is entirely based on the characteristics of the R group, because as we know, there's nothing else that's left for variability. The other groups are pre-decided, the chirality is pre-decided, except in one case. We'll get to that in a bit. So everything is kind of fixed, even the length of the chain is decided. So it has to be an alpha amino acid. Only one carbon between the acidic group and the amino group. So this is the only place where there can be variability, is in the structure of the R group. For non-polar amino acids, the R group has no dipole moment or relatively little dipole moment. Polar amino acids have a significant dipole moment in the R group. Acidic amino acids have an extra COOH in the R group, like our friend aspartate, whom we studied before. And basic amino acids have an extra NH2 or some other NH bearing group in the R group, which allows them to be which allows them to acquire a doubly cationic form. And these guys can acquire a doubly anionic form. So let's look at them in some detail. Obviously, you don't need to know all 20 of them. One thing I'd advise you to check, which you'll need to revise right before your exams, is which are essential, which are unessential. I'll come to that at the end of this section. Okay, let's look at the non-polar amino acids. The first is alanine. Alanine is very simple. It just has a methyl group as its R group. Okay. It is written in short as ALA, A-L-A. Okay. And sometimes there are specific cases where we need to save it as a very, very short code indeed. Then we just use the letter A. We prefer to use the 3-letter codes because they're obvious. They're generally just the first 3 letters of each amino acid, with only I think 2 or 3 exceptions. The first 3 letters of the amino acid are basically the name of the amino acid itself. But the 1-letter codes are also available and they are used sometimes. Less common, but they are used. So alanine, denoted as ALA or A, has the R group as a simple methyl group. Phenylalanine, which I've written in yellow because it's an aromatic amino acid, we'll come to aromatic amino acids specifically in a bit, is the same CH3, but we've substituted one of the H's with a PH. So this is one of those examples. The 3-letter code is PHE, but you can't just call it P because P is the code for proline. So we write F over here as a single letter code. Then you have valine, which is C. It comes to CH over here, and then it has a CH3 and a CH3 on both sides. So it's got a branched position at the first carbon in the R group has got 2 methyl groups attached to it. Okay, this is interesting, and we'll come back to valine later. Now we come to glycine. Glycine is an interesting amino acid because it is the only achiral amino acid. Its R group is a simple H, just a hydrogen. So it has 2 hydrogens on it. It's achiral, and that gives it extraordinary flexibility. Okay, when every single amino acid is of a particular chirality, then they must be organized in a particular way so that R groups don't hit each other. But whenever you have a section of glycines, they can organize themselves in any random way. So very flexible proteins will sometimes have a stretch of this glycine. These are called polyglycine L-alpha helices. We'll get to that in a bit. But basically, it's the most flexible. It doesn't have to worry about anything. So you should definitely remember glycine, at least if not the previous three I've mentioned. Okay. Then comes methionine. Methionine is another must remember amino acid. Because in any protein, and we'll get to this in a bit, but just like our sugars had two terminals, right? One which had a free C4 end and one had a free C1 end, right? Either alpha or beta. Similarly, proteins will have, when you look at a protein, what is the structure of a protein? Protein structure is something like this, right? Remember, I discussed the peptide bond at the beginning of this class. So then you have NH, and then you have C, and then you have R2. So this is just a dipeptide. Dipeptide means it's made out of two different amino acids, right? So what you can see that this end will have NH2, right? It's not formed an amide. This H has not been consumed, and this end will have a free COOH. Only in the middle they formed an amide. Now if I make it extremely long, there will be many more amides in the middle. If I add say 100 amino acids, 1000 amino acids, but the ends will always have this, which is called the N terminal because there's nitrogen, and this which is called the C terminal or the carboxyl terminal. So methionine is found at the N terminal of all proteins. When a protein is synthesized, we club a whole bunch of amino acids together, and the first amino acid which triggers the synthetic process, which we will study in a few videos time, the first amino acid is methionine. Methionine has been written in green color because it's a sulfur containing amino acid. You can see it's what we call a thioether. Like an ether which has an oxygen in the middle of the group, this has a sulfur in the middle of the group. That's why it's called a thioether. And it's a very important amino acid, so we should remember it also. Any sulfur containing amino acids, they'll give a very bad smell when burnt. We'll get to that in a bit. Then we have leucine, which is just another one of these non-polar. So these non-polar ones, you can see they're mostly just aromatic chains or aliphatic chains. They're just hydrocarbons. This one is an exception because there's a sulfur in the middle, but still the dipole moment is relatively low, so it is classified as non-polar. If I go to leucine, it's something like this. Isoleucine, again it's a similar chain. We've just moved one of the branches one carbon back. But like valine, that means that at the second carbon, it's got a branch. We'll come back to this property. So remember, valine, isoleucine, you don't have to remember the names, but remember there are some amino acids which are branched at the second carbon. By second carbon, I am referring to the first carbon of the R group. This is the first carbon. So the first carbon of this group, the R group, is the second carbon. When I say branch of the second carbon, I mean branch of the first carbon of the R group. This is the first carbon of the whole chain. Okay. So this is branch of the second carbon, this is branch of the second carbon. Remember this property, we'll come back to it in a bit. Then we have proline. Proline is another must remember amino acid. So far, glycine you must remember, methionine you must remember, proline you should definitely remember. Okay. Proline, abbreviated pro or P, is really weird and cool because, so let's say this is the first carbon which is part of the main chain. This is the first carbon of the R group. And it goes back in a pentagon and it links back to this N terminal. So what's basically happened is as if this R group is this chain that comes and joins here and one of the H's is missing. So it's a really cool sort of chain, and it's this one chain that folds backwards. Most R groups will just stick out, but this R group is bent backwards. This is trying to meet up with the NH2, and therefore this is technically called an amino acid. Not an amino acid, but an amino acid. Because the cyclic amine is also called an imine, right? And this is going to be very important. Proline also has some pretty interesting properties. A great deal of, just like glycine is the most free amino acid in terms of rotation, proline is the most rigid. Because it's bent back in this manner, it has very limited degrees of freedom when it comes to rotation. So this is an amino acid. Then we come to the polar amino acids. Polar amino acids, one is serine, which is CH2OH. Serine you should remember, not because it is of high importance, well it is all the amino acids of high importance, not because it is any outstanding feature in this particular part of our syllabus, but just because we used it to define the DNN configuration. So you should remember, serine is the one that has CH2OH. Threonine is like serine, Just like valine or isoleucine, it has got this extra branch. So it's branched at the second carbon. Okay, so the next amino acid we have to discuss is tryptophan. You can see I've written it in yellow over here because again it is an aromatic amino acid. It's made out of a ring known as an indole ring. Okay, so that's basically this sort of pentagonal ring linked with a common edge to this hexagonal ring, both of which are aromatic, and such that this particular vertex is a nitrogen. Okay, now tryptophan is very interesting and very important because when you've got a sample from some organism and you want to see how much of whether it is protein, say this is a very common thing we do. To try and get an understanding of what the structure of a particular molecule is, we crystallize it. Okay, we take out a sample from an organism and we crystallize it. Then we put the crystal into an X-ray machine, not an X-ray machine, sorry, what's called an X-ray crystallography machine. We shoot it with X-rays. The X-rays will diffract around individual atoms, and from the diffraction pattern we will know which atom was where. So it's very important that when you take these crystals, you should put the protein crystal through the diffraction and not some crystal of some salt which is also inside that living organism cells. So how will you know which one belongs to a protein and which one is of a salt? So the trick is you put whatever you want under a UV light. Tryptophan is such that because it's aromatic, it will absorb whatever UV light you give and its electrons will be promoted from the bonding to the anti-bonding molecular orbital. But since there are so many more bonding molecular orbitals than anti-bonding molecular orbitals, it will not break into pieces. Because what is the logic of an aromatic system is that the maximum number of electrons are in bonding molecular orbitals. So it will get promoted to anti-bonding molecular orbital and then immediately get demoted and it will give this bright white flash. This is true of any of the aromatic amino acids, all the ones which are in yellow, but this one gives the best result. So you can see that under a microscope, if you flash it with UV, it will flash back in white color. And so you know that this whatever is there, it has tryptophan in it, and so it must be protein and not a salt. So this is a very useful application of tryptophan. Then you have tyrosine, which is just phenylalanine with this OH added over here. So tyrosine and histidine are two amino acids which again are aromatic in nature. Histidine, you should see it is aromatic. It's quite confusing. This is the chain. So first a carbon, then this cycle which has two nitrogens in it. So if you see, there is this double bond here. The lone pair of this nitrogen and double bond here. So this nitrogen's lone pair is not involved in resonance. Therefore, though it is classified as one of the polar ones, in certain circumstances, it can easily switch to being one of the basic ones because this nitrogen which has a lone pair can accept a H+, and this allows histidine to have some phenomenal diversity in terms of where it can occur in your protein. It can occur deep inside the protein when it is uncharged and so it is surrounded by other fatty parts of the protein, hydrophobic parts of the protein, or it can acquire a charge and it will be exposed to the surface because it will be pulled to the surface of the protein by water molecules. And because it can switch from being charged to uncharged, it can be part of what is called a charge transfer relay, which is when a protein wants to extract some charge from a place to another place. So you can have a relay of histidines which will quickly become charged, uncharged, and they'll basically play passing the parcel with a proton, and you can move a proton from one place to another. Many proteins actually use this technique to move protons. Okay, so histidine is also very important. But this function I told you is not mentioned in syllabus. Even for tryptophan, the function I told you is not mentioned in syllabus. So while glycine and methionine and proline you should definitely remember, here maybe I shouldn't have marked it with an asterisk. You don't need to remember it. It's not essential to know. The next one is cysteine. This is essential to know even within your syllabus. You should know its name. You should know its properties. The other ones, they're good to know. You can forget them, the ones that I've explained. But this you should definitely not forget. This is like methionine, but instead of being a thioether, it is just a thiol. The SH in methionine gets replaced with SCH3. Here it remains SH. Cysteine is denoted by CYS or C. Okay. And the cool thing about cysteine and cysteine's property is that it can form a per-sulfide or a disulfide bridge or a disulfide bond. So let's see what I mean by that. What I mean is you should understand something about the environment of inside a cell versus outside a cell. Inside a cell we have a reducing environment. Many reductants, good reducing agents are floating freely inside the cell. So cysteine will tend to exist as the R group will tend to exist like this, CS2SH. But when it goes outside the cell, if there is a protein that is between cells or in an acellular region, then if there is another cysteine somewhere else, then what's going to happen, right, is that since outside the cell you have an oxidative environment, some oxidative agent will remove these hydrogens. As we know, one definition of oxidation is the removal of hydrogen. Some freely floating oxidating reagent will remove these hydrogens, and these sulfur molecules will be directly linked by a bond. Now we know that this also happens for oxygen, as in peroxides, right? Like hydrogen peroxide. But persulfides or disulfides are a lot more stable than peroxides because of the larger atomic radius. So this is a stable persulfide. Now what happens is, you can have a large fat protein which has these cysteines at random parts of the protein, and the protein will get folded up and joined up together because these cysteines will start forming these disulfide bridges among themselves. On this protein I could have some protein which has a chain like this, but here I might have a cysteine and here I might have a cysteine, and as soon as the protein exits the cell, it will enter an oxidative environment and they will be bonded together by a disulfide bridge. This helps give protein certain strength. Very strong, tough, fibrous proteins generally have disulfide bridges, which means they have a high cysteine content. And a good example is your hair, keratin in the hair and in the nails. Coming back to what I mentioned when we were talking about methionine also, these are sulfur-containing amino acids. When they burn, they leave a bad smell. So that smell you smell when hair is being burnt. If you've ever seen hair being burnt, or rather smelt it, it has this really terrible smell, right? That's because there's burning sulfur in those particular amino acids. So don't forget cysteine, methionine, glycine, or proline. So far, now asparagine and glutamine are very similar to each other. Asparagine has a methyl group and then an amide. Glutamine has an ethyl group and then an amide. So asparagine and glutamine, because they are amides, have this very interesting property. That is, when plants, so you will have studied in lower classes, like from kindergarten onwards, maybe not kindergarten, from some middle school onwards, that if you grow leguminous plants, okay, in soil, then there will be a high amount of nitrogen in the soil when these plants die and you plow it over. How does that happen? Leguminous plants have some bacteria called rhizobacteria that live on them, which will fix this nitrogen. But actually, rhizobacteria will convert nitrogen into ammonia, which, as you hopefully know, is toxic, right? Ammonia is toxic. So the plants have quite a deal of difficulty. So actually, this ammonia is further converted into glutamine and asparagine. They always convert it in the forms of these two amino acids. So forget about proteins. One use of amino acids is to create proteins. These two amino acids are used in proteins, but they are also used by plants as just stores of nitrogen. Okay, and then when your leguminous plants die and you plow the earth, then these amino acids get released back into soil and they can be broken down into some other compounds, and then your actual crop will take it up from them. So this is how the nitrogen is stored in these amidic amino acids. One thing I should mention is that serine and threonine, they can sometimes form hydrogen bonding among themselves. That can also lead to a certain amount of stability. Just like cysteine can form a covalent bond, disulfide bond, serine and threonine, they can form hydrogen bonds between these OHs. Okay. Next we come to the acidic. So the acidic ones are really easy. If you know asparagine and glutamine, you convert these amides into carboxylic acids, you get aspartate and glutamate, which are represented by D and E. We can't take A and G because A is alanine and G is glycine. So we go by D and E. Okay, and these are acidic amino acids. We already studied aspartate. So you can get questions based on aspartate and glutamate where you will have the Ka1, Ka2 and Ka3 value, and you have to do that thing I told you. I hope you remember that. You can always review that part of the video if you forgot. Now the interesting one, basic amino acids. Lysine is very simple. It's just a long carbon chain ending in NH2. So if you understand how an acidic amino acid works, it's pretty much the same. But arginine does not just have an extra NH2. It has this interesting group over here called a guanidinium group. Okay, and here's what happens. This nitrogen has a lone pair of electrons, a double bond and a single bond. Say an H plus comes over here. Okay, so this nitrogen will become positively charged and it will have this double bond over here. Right? Why is it positively charged? This H plus came and pulled the lone pair into a covalent bond. Now what will happen is, this nitrogen has a double bond, has a lone pair of electrons. It can donate this lone pair of electrons here. So it will acquire a positive charge and double bond will form over here. Now this carbon will say, I don't need so many bonds, I can't be pentavalent. So this bond gets released and both the electrons in this bond will come up here and form a lone pair of electrons. Similarly, this NH2 can donate its electrons here. So what ultimately ends up happening is you have what is known as a Y-aromatic system. This is not a proper aromatic system. It's similar to CO2 whole minus. In CO2 whole minus also, the molecule or the ion is Y-shaped and the electrons are delocalized. So just like cyclic systems or properly aromatic systems have a lot of stability, Y-aromatic systems are also quite stable. Okay, so it won't actually be like a Y, it will be at a perfect 120-degree angle everywhere. And this positive charge will be distributed over all three. So it is resonant, so it has got a very stable, it's very stable even when it's positively charged. Also, generally if you have like aspartame and glutamate, no, you can replace one with the other. Same protein, same function, just replace glutamate with aspartame, there won't be too much difference in how it behaves. This is also true with lysine and arginine because the main function is to acquire a extra to be doubly positively charged. So that doubly positive charge will help the protein in its structure and its function by repelling some group or by attracting some group. But arginine cannot always be replaced with lysine, whereas these two are generally always mutually replaceable. This is not the case. Arginine, because of all of these extra hydrogens, it can form a lot of hydrogen bonds with phosphate groups. So you studied, for example, how ADP becomes ATP, or rather how AMP becomes ADP, which further becomes ATP. So there are enzymes made out of proteins which are catching these phosphate groups and attaching it to the ADP or to the AMP. And then there will be other enzymes which remove these P groups. And arginine is very good at that. Okay, it's very good at enzymes that remove these phosphate groups.